in the precision medicine program at the University of Edinburgh. And I'm based at the Institute of Genetics and Cancer, where I'm supervised by Professor Wendy and Dr. Simon Beatty. Today, I will present our work, which is now out as a preprint, focusing on benchmarking tissue and cell type of origin deconvolution in cell-free RNA. I will start with a brief introduction to cell-free RNA. What is it and why we need to deconvolve it in the first place? Then I will walk you through the benchmarking we designed and how we applied it into both simulations as well as validated in real-world cell-free RNA datasets, and then summarize the key limitations, findings, and future directions. So cell-free RNA consists of mRNA molecules that circulate in human biofluids, such as the blood and the urine. They are present at very low levels in healthy individuals and are highly degraded. However, during diseases, these contributions seem to increase their levels because there are a lot of contributions from also solid organs, solid tissues, which make cell-free RNA a powerful tool for non-invasive monitoring, such as tissue biopsies or in other different contexts. Essentially, cell-free RNA is an analyte that originates from many biological processes. It's not from a single source or a single mechanism. It is released through a number of different active and passive ways, such as during cell death or necrosis, or active ways such as extracellular vesicles or cell-to-cell communication. And essentially, when we observe cell-free RNA, it's not a clean signal. It's a mixture of contributions from different tissues and processes. And because cell-free RNA originates from many sources, it can capture signals from a wide range of conditions. For example, there is evidence of use of cell-free RNA in different malignancies, cancer patients, in different pregnancy-related syndromes, as well as in infections and in donor-derived nucleic acids in transplantation, which makes it a powerful, minimally invasive biomarker. And one seminal study in the field of cell-free RNA was this particular one, as in this study, they focus on patients with preeclampsia. Preeclampsia is a multi-organ pregnancy-related syndrome that can lead to organ failure. When they assess the differentially expressed genes in these patients with these conditions, they see that 13% of them were either tissue or cell type specific. And because of this specificity of some transcripts, they were able to longitudinally monitor these transcripts over time to assess the organ health of the pregnant mother, as well as the risk of developing organ injury, which highlights the key idea that the signals can come from remote tissues and the blood is like a readout of the whole body. So more recent approaches of analyzing cell-free RNA use more advanced ways known as deconvolution approaches. Traditionally, deconvolution infers cell type proportions from bulk transcriptomes. So we start from a single bulk measurement, such as a human brain or a human liver sample. Then we apply one of the many deconvolution methods alongside a reference matrix in order to get the proportions of each cell type that are present in that sample. So there is a variety of deconvolution methods available, and typically use a reference gene expression profile of the cell types that we expect to see in that sample. Such as when we go from bulk brain, we will go to the different the brain cell types that we could see there. And there have been different deconvolution approaches done in the past in different contexts. One main point is that the convolution algorithms and methods were developed for going from the bulk tissue, as I said, to the cell types that are present there. This means that the number of potential contributors, usually in deconvolution, is much lower than what you would expect in cell-free RNA. So in the convolution in bulks, conditions usually goes from 10 to 20 cell types. And in these studies that are summarized from this review, essentially test the deconvolution in different application and different context. And essentially, this context ranges from human immune complex or the human brain. And when they test the different deconvolution approaches and the different subsequent references, we see that the performance depends on both of them. So then comes to the question, so what about cell-free RNA? Because cell-free RNA is indeed a mixed signal and contains contributions from many more tissues or cell types than that typical deconvolutions will expect. And on top of that, signals are very fragmented, sparse, and noisy. As a result, when we generate sequencing libraries from the sample, we get a lot of duplicates. So it's not so good, not so good sample, which make inference fundamentally more difficult and challenging. So in liquid biopsy, there have been applications of deconvolution, and typically they use the Tabula Sapiens Atlas, which you can see schematic here, and one of the many available tools. However, they do that without much systemic evaluation of the tools and the reference in these settings. And importantly, from the schematic, you can see that even though this is the most comprehensive atlas we have available and contains a number of different organs, even from the same donors, we see that this atlas lacks the human brain, which can be important for some cases. And when we eventually apply the deconvolution of cell-free RNA, the output we get is something like this. This is an example from healthy individuals. So in this case, we see that the majority of the signal, of the proportions, of the estimated proportions, come from blood cells, because there is not much coming from the solid tissues. However, then the other issue is that this reference atlas that is mostly used is incomplete and lacks the brain's specific expression profiles. And when the researchers have applied this atlas with one of the many deconvolution approaches into bulk brain data, we see that the signal is misassigned to closely related cell types, such as Schwann cells or kidney epithelial cells, suggesting that there is a spillover and loss of resolution of accuracy. So overall, there are three main gaps in cell-free RNA deconvolution. First of all, the convolution methods have not been benchmarked, compared or evaluated in these more complex multi-organ settings where there is many more potential contributors. The other thing is that the widely used cell type reference lack key tissues such as brain. And lastly, we see that tissue of origin is generally not widely used in the literature and definitely has not been benchmarked in the literature. And this for us was a bit surprising, because in the tissue level deconvolution, you will go into a few dozens of potential organs, potential tissues, instead of in the cell type deconvolution, where the potential contributors can be more than 100. So overall, based on this, we designed a benchmark for cell-free RNA deconvolution. We generated reference profile, custom reference profiles, and we did it separately for both tissue and cell type of origin. For the tissue of origin, we use publicly available GTEx data, where we did some more handling that I will explain later. While for the cell type of origin, we use Tabula Sapiens version 1 that we augmented with two distinct brain datasets. And for to test them, we also needed to have a ground truth known proportions. So for that reason, we generated simulations, which is what is typically done in benchmarking studies. So in the tissue level, we generated tissue mixtures by focusing on atlases that have multiple tissues from the same individual, and we mix these tissues together. And the cell type level, again, in a similar manner, we aggregated single cells from the same individual, and then we added perturbations such as noise and removing rapidly degrading genes to make this simulation less ideal case scenario, to make it much more to cell-free RNA. And we applied eventually to 7 deconvolution methods. I will explain these methods later, but we selected these 7 methods because they perform better in previous less complex benchmarks, or because they are widely used in liquid biopsy studies. So since it's a computational problem, we needed some good metrics to assess the performance. And we use common deconvolution metrics, essentially. So these are mean absolute error and absolute error. And we also use Pearson correlation, and doing it also absolute error per tissue or per cell type. Essentially, these metrics are used in many deconvolution studies. And what they compare the known proportions, the ground truth of the simulations, versus the inferred, the predicted proportions, to see which method and reference works better, is more accurate, essentially. And then as I said, we assess 7 deconvolution methods. These 7 methods, they are listed here, and they span 3 different reference types. So this is our categorization, so it's not widely used in the literature, but we categorize them into matrix-based, which essentially use a pre-construct matrix as their reference in order to identify the specific genes or the specific features of each cell or tissue. A reference base, which takes the whole expression profile as the input matrix, and a signature base, which it takes the full expression profile, but also takes a user-defined number of signatures to use per contributor, and builds essentially the matrix each time on the fly. And these tools span 2 different categories. The top panel here, you see there are custom tools. So essentially, they are implementations of algorithms, mostly using Python. While in the bottom panel, you can see these are published approaches. So it's tools that have been out and can be more directly applied, let's say. And overall, these span different algorithmic frameworks. Some are regression-based, such as machine learning, support vector regression. Some are Bayesian, some are probabilistic models, and some are more linear models. So for the tissue of origin, this is a bit more detailed schematic of what we did. So starting from the GTEx V8 counts, we merged transcriptionally similar tissues together, and this was done to decrease the number of potential contributors and the complexity of our problem. And then we sample counts, which is the features that are used as a reference for the deconvolution, using 3 different approaches. And as I said before, we generated simulated samples. We use this atlas that has many different organs coming from healthy individuals. And we mix expression data from these organs and tissues from the same donors in different proportions. And we did the perturbation as well, and then we applied the deconvolution using the different methods we identify, using the different inputs that each method wants. So, time for some results. Essentially, in the tissue level, we use the 7 deconvolution methods with 9 reference matrices that come from 3 different sources. So for this, we tested it with 1,000 tissue mixtures with random proportions. And I'm showing here the 2 metrics that I explained before, which is mean absolute error and Pearson correlation. Overall, we saw agreement between the 2 metrics. Just to be clear, the mean absolute error, essentially lower is better, and Pearson correlation, a value closer to 1 is better. So we see that some particular combinations, such as Bayes prism with the central reference, or Reedy-Conv with the random 10 reference, seems to work better than others. And then because we had these mixtures of simulation, we're also able to see the performance at the tissue level. Essentially, we could ask the question whether the performance error is similar across tissues or whether some tissues are harder to resolve. And when we assess this, we saw that actually some tissues are harder to resolve, and some tissues such as kidney, liver, lung show consistently higher error across the methods. And we see that this effect is actually more pronounced in a specific category of the deconvolution tools, those that are matrix-based methods. So those that they need a predefined signature of matrices to work. And overall, I will not show the result just in the interest of time, but this is shown in the preprint that we found that there are a number of different failure modes in tissue level deconvolution, similar to what was previously reported for cell type deconvolution. Overall, we saw that some tools overestimate absent tissues. So there are a subset of simulations had by design lacking some organs. So in this, we see that when the tissue is truly absent, we still get non-zero predictions, suggesting some model bias or some model inaccuracies. Then we also see that the transcriptionally similar tissues were more prone to being misassigned or spill over into another category, essentially.
Alex,
And we also, yes?
on the previous slides, I think we're presenting results on the mean absolute error, and I'm wondering, like, since like the tissue of origin performance is good in a way, and what could be the interpretation, like what does good performance mean for the tissue of origin in this deconvolution process? Like,
You mean what value? What would be a good threshold, essentially?
not really. Like,
Mm,
if the perform like what is bad performance, what is good performance in the deconvolution sense, but when it comes to the tissue of origin. But also, I don't know whether that question is clear, but also somewhere we talked about cell of origin. Some of us are not fundamentally from biology, so
yeah,
somewhere we get mixed into some of the biological terms. But I'm drawing my attention to the tissue of origin for now.
yes,
But I want to get an understanding of the good performance when it comes to the mean absolute error.
yes.
What interpretation can be taken away from better performance in terms of deconvolution?
Okay, when there is a low mean absolute error in this case, essentially the tool, the method and reference combination, they accurately predict the proportion as it is. So, uh, and then it means that a small absolute error, mean absolute error, is that organ that organ is accurately inferred, accurately predicted. That is match our ground truth, our known proportions. Is clear?
Okay, yes. And then does that mean that in the process of deconvolution, we can determine where like the organs, the independent organs within the body, like what we are seeing here, like we are seeing female reproductive and the tibial nerve
Mm-hmm.
giving comparatively better sort of performance when it comes to the mean absolute error.
Oh,
So
no,
my
they give comparatively worse because it's quite higher. So the
oh yeah,
female reproductive is one of the most problematic we got.
okay, yes, sorry, yes, so uh does
But
that mean that in the
yeah,
deconvolution we can determine where the cells are coming from in that sense, but also uh how do we like separate them and then attach the different organs to the different sort of like So should I say tissues or cells within maybe the sample that is being deconvolved?
Yes. So just to clarify, when we assess tissue level, tissue of origin, it's only tissue. We don't do anything about the cells. So yes, cells is not
Okay.
directly relevant to tissue of origin. Essentially, what it means in practical terms is that we get higher error in these tissues. So if we try to deconvolve the sample of a patient that has damage in the tibial nerve on the female reproductive, then the values that we get may not be so trustworthy compared to others that are much more accurate. But also, of course it's dependent on the simulations that we use, because it's a limitation of the availability of data, that when
Okay.
because we wanted to have from the same individual the different organs to match more cell-free RNA. So it was not possible to have all the tissues that we show here. And another parameter affecting this, so because GTEx has 54 tissues, so 54 organs or 54 regions of organs. And to decrease the complexity of our problem, we merge some that are quite relatively similar together, or they have either a transcriptionally similar profile, or they have a biological use that is similar. So for that reason, the female reproductive was merged, but essentially it contains a number of different organs. But because it was not really crucial for our analysis and our future clinical studies, that's one of the reasons we merged it. But again, each tissue performance is also dependent on the number of samples, on the variability between patients and cohorts. So so it's not so dependent on one organ being less difficult to understand and decipher. It's more about based on what is available at the moment.
All right.
Yeah,
Thank
so
you. I
yes.
I think that is great. The subsequent questions, I think I'll wait for you to have full presentation of results, because I have questions about the Bayes prism giving
Yes.
better performance, but I think it will come at the end of the session. Thank you.
Okay, yes. So did I finish this slide? Just to confirm?
I think you were on systematic bias.
So the reference, we saw that how the reference was constructed essentially affected the convolution as much as changing between algorithms. And then this is the cell type of origin design. So from now on, when I talk about simulations, I will talk about the cell type level. So essentially, as I said, we mix the Tabula Sapiens version 1 with two different ways using two different brain datasets. Those are human single-cell brain datasets. So they were mixed or augmented with the Tabula Sapiens multi-organ single-cell dataset. And then we took Tabula Sapiens version 2, which is different donors and different batch, and we use that to generate our pseudo bulks, which is single cell aggregates. So essentially, we add the expression together instead of doing it at the tissue. So instead of mixing tissues, we mix individual cells, so that in our simulation we can know the proportion of different cells. So we can know that our simulation has 10 B cells or 20 T cells, and go on and go on. And so again, we did the same noise addition and gene removal as I described before, and I applied the cell type deconvolution. So here we see a bit different results compared to before. We see that based on the three different references that we used, we see that the performance much more, and we see that the ranking of methods across the different references seems to be maintained. But same as before, we see that Bayes prism and redeconv give the best results by having a lower mean absolute error and a higher Pearson correlation. And then we see that also CyberSort is a good tool in this case. And then we also assess similar to the per tissue level, we're able to assess the performance in the per cell type level. So we check the 10 cell types that were present in our simulations and had the highest error. So they are, if on average they had 10% known ground truth, the tools might have predicted 20 overall. So this is a mean absolute error of 10. And so we assess the 10 cell types with the highest. And overall, we see that specific cell types have higher errors. And we see that can be mostly assigned to immune populations, potentially due to the number of cell types that were merged. And we see that there's also sensitivities between methods and references. But overall, even though I showed the results for one of the references in the preprint and in general, when we compare the three references, we see that it's very similar, the picture between them. And in the introduction, I also pointed out that one of the main reasons that we augmented brain is because brain was absent from the tabula sapiens, and when researchers deconvolve bulk data from the brain, they get other cell types being assigned. So in our case, we deconvolve brain using different references that included brain datasets, single cell brain datasets. And we see that now we are able to detect a high percentage of brain cell type. We see that there is quite variability between the tools, and especially, which means that especially for music and Redecon, we see that there is preference due to the model architecture in one of the references. But essentially, it shows that in the missing when brain was missing from the reference, we got false signal, but now with the augmentation, we can capture a more accurate signal. And next, we wanted to evaluate how tissue and cell type perform in less ideal datasets, less ideal simulations. So to do that, here I'm showing only tissue of origin. To do that, essentially, we added in the top panel, we added noise. We added negative binomial noise to play with the counts and make it tougher for the tools to characterize. So essentially, instead of to not be as ideal case scenario as before. So in the top panel, using the same metrics as before, we can see that Redeconf and Bias Prism showed quite stable performance, while the other tools seems to become worse. And then in the bottom panel, I'm showing a completely different robustness test. So since it's cell-free RNA, it's highly fragmented. And however, mRNA in the cells, it also can be fragmented and has a different rate of degradation, something we call in biology half-life. So because we saw that in cell-free RNA datasets and in general other bulk expression datasets, there is higher detection of more stable transcripts. So we hypothesize that if the convolution depends on faster degrading transcripts, then it's possible that the performance could be worse. So in the tissue level, we remove deciles, so 10%, 20%, 30% of these rapidly degrading genes based on the current estimations of half-lives. And we see that in the tissue level, does not seem to have a much impact. Especially, we see that Bayes Prism actually improves, potentially due to the lower complexity of the library. But then we also did the same test in the cell type of origin. So in the cell type of origin, we see that in the noise level, when we add higher levels of noise, again, the performance becomes worse, as we expected. But some tools, again, are more stable, such as Redeconf and Bayes Prism. While in the bottom panel, you can see that when we remove rapidly degrading genes, as I explained before, we see that the performance of the tools becomes worse across all tools. So essentially suggesting that the tissue and the cell type of origin have different robustness profiles. And this decrease in performance and accuracy can be because of dependence on specific signatures or finer grain resolution of the cell type deconvolution. And lastly, a big part of the paper and the study was essentially to test it also to real world data, because the simulations are just a model, and at the end of the day, we need to see it in real world data how this performs. And there is a plethora of clinical conditions that can be characterized and published for cell-free RNA that are available for researchers. However, the tricky part when using published cell-free RNA datasets compared to before, where it was controlled simulation settings, is that for published datasets, we cannot know the ground truth. We cannot know the actual proportions of tissues. So in that case, we need to become more innovative to do the different type of comparisons. So in the different data sets, what we did is we compare the inference, so the computational output, with the biochemical markers of injury. So this is basically we correlated them using how our tools would detect organ injury based on cell-free RNA versus how it's currently done in the hospitals, in the clinic. And then we also did comparisons between cohorts, such as diseased versus healthy individuals. I will show a few examples for that, and I will show both the tissue level and then the cell type level. So in the tissue level, we see that we applied it into an acute pediatric cohort, which was taken from this paper. This cohort had much measurements of ALT. ALT is a protein that is specific to the liver, and in the clinic, in the hospital, is a biochemical marker of liver injury. So we compare our inference, our predictions of liver with the current gold standard, essentially. And we see that the Bayes Prince gives the highest correlation. And we also see that other tools give similar correlations around 0.6, such as CyberSort, Music, and Ridicon, which suggests that tissue-level cell-free RNA can reflect biochemical liver injury. And then we also did a sort of different comparison that is done widely in the literatures. So I'm showing here two different patient cohorts. The first one here is the acute cohort, which is the same as before. And what we compare here is we compare the liver contributions between two different groups of patients. These patients were categorized based on the ALT values that we have, whether they had normal ALT or high ALT, based on what they were defined in the published study. And overall, we see that the majority of the tools detect statistically significant difference between these categories. However, we can see that not all tools do, and we can also see that the magnitude of inference is quite variable between the tools. And in the second cohort, we compare a chronic liver cohort, so a cohort that has chronic disease associated with the liver damage, essentially. And when we compare disease versus healthy individuals, again, we see the same pattern that in that case, all tools detect statistically significance, but with different rates. And we also see that the magnitude of how much liver each tool predicts is different. And then we go into the cell type deconvolution. So in the cell type reference, our the cell type that will match ALT measurements in theory is hepatocytes, because it's a liver-specific cell that is widely used in, that is specific to the liver, essentially. And when we correlate the hepatocyte contributions across the different tools with the ALT, similar to what we did for the tissue, we see that, again, the best tool is Bayes prism, as it gives a 0.6 Spearman correlation value. But then we see that the other top tools now decrease a lot, which suggesting that fewer methods show consistent associations, which contradicts what we see for the tissue level, and suggests that the cell type inference could be less robust and less reliable in this setting. And since we had the groups of patients with high and normal ALT in this acute liver cohort, we also compared to see if we will get statistically significant associations between expected cell types. So here I'm showing the 7 different tools and a number of different cell types that came out statistically significant. Overall, we would have expected hepatocyte to be statistically significant between this cohort, these groups, these patient groups. But we see that it's only detected by Bayes prism. And overall, we see that the cell types are not consistently detected across tools, it's more sparse, which suggests that inference is not only weaker, but could also be less reproducible across methods for this cohort. And another example that we wanted to test is whether the brain-augmented references could detect additional resolution compared to the published studies that used only the tabula sapiens reference. So in this case, essentially, we used a cohort that had healthy individuals, controls. We had COVID-19 patients, and they had patients with multi-system inflammatory syndrome in children. This is also known as MISC, and MISC is a severe complication of COVID where we had different body parts could become inflated. And on the first panel, we can see the results from the original study. The authors show that there is higher levels of Schwann cells in the MISC group, compared to COVID, but also compared to controls. However, since we know that the tabula sapiens lack the brain, and when there is a bulk brain deconvolution, signal could be misassigned to Schwann cells, which is neuron cells, but of the periphery, not of the brain. We applied the brain-augmented references to the same patients. Overall, we see that all of our tools detect neuronal signal across all methods with statistical significance. And what we see at the same time is that Schwann cell signal become much less prominent and is only detected by very few tools, which suggests that it shows that it's important to have as many tissues and cell types as possible in the reference, because the misassignment of the signal could show a different closely associated cell type as significant. And the last result slide is, I wanted to show it's a method-dependent variability in cell type inference against cell type deconvolution, but in an Alzheimer cohort. So here we compare Alzheimer patients with controls, and we see that across the different tools, we detect a number of neuronal and brain cell types that again would have been missed if we didn't augment the brain data sets. And we see cells such as glial, astrocytes, microglia, neurons, and other immune cell types to come up statistically significant, potentially showing signs of neuroinflammation and neurodegenerative processes. So again, showing that in this example, the detection was much more stable across the tools, but again, there were problematic tools and differences between the different tools and reference. So overall, this is a joint benchmark of tissue and cell type deconvolution in a body-wide setting. And one of the key results is not the convolution performance was not impacted by the method alone, but it was also dependent strongly on the method and the reference. And we saw that in many cases, when we changed the reference or how the reference was generated, we could get results as much result difference or accuracy difference as much as changing between different deconvolution tools. And another important observation is that tissue level inference was more consistent with the clinical markers of organ injury. And overall, this study suggests that the convolution can recover meaningful signals from cell-free RNA, but the performance is very variable and must be interpreted very carefully. And we see that absolute proportions are method dependent. So essentially one tool could predict 2%, but the other tool could predict 10%. But if you compare within the same tool the patient cohorts, we see that it's more accurate. So essentially any conclusions or interpretations will be done by comparing within the same tool, not by setting a threshold, for instance. And we see that also that the cell-free RNA profiles and properties such as the fragmentation, it introduces an additional challenge that should be taken into account in the future or should be used as a motivation for generation of cell-free RNA degradation models, or in general RNA degradation models. And then we also see that it's very important that all the reference should be complete for cell-free RNA deconvolution, and which matches what we saw, what was reported in the literature for less complex deconvolution benchmarks. And the tissues and the cell types that the tools will predict will depend on that. And we also see an interesting resolution versus reliability trade-off, which was a bit expected. But we see that the tissue level is more robust and consistent across the methods and show better agreements with the current gold standard of biochemical markers. But we also see that cell type level deconvolution, so going to the specific types of cells, can give higher resolution, but is less stable across the methods and the references, and but shows also greater variability. So essentially, when we increase granularity on the deconvolution setting, we decrease the identifiability of the cells. And in the future directions, also some important directions that could improve cell-free RNA deconvolution. And from a data perspective, they could be more complete and diverse reference atlases are essentials. And this can also be not only more tissues or more cell types, it can also be more disease states, as we know that expression is impacted by the pathological condition of the cell type, such as stress. Cell types are slightly different, their expression, than normal healthy cell types. And from a modeling perspective, there is a need for methods to account for cell-free RNA properties, such as the fragmentation or the degradation that we expect in this analyte. And if we manage to incorporate these factors into the model, it can be even more accurate. So it's an opportunity for computational development, essentially. And lastly, it can also be an opportunity for developing methods that are more suited for high-dimensional mixtures. So these more complex scenarios or even missing features, which are inherent for cell-free RNA. And improving both the data and the modeling it will be key to make cell-free RNA deconvolution more reliable and more useful in clinical settings. And overall, I would like to thank you all for your time. I would like to thank my supervisors, which is Professor Wendy Bickmore, Dr. Simon Bidi, and Professor Dowd at Karolinska Institutet. And I would like to thank the Medical Research Council UK and the University of Edinburgh for funding my PhD, all the members of the Bigmore Lab, and here you can see a QR code of the preprint in case you want to read something more or something more detailed.
Thank you. Thank you, Alex. My first question is about the the tissue cell robust performance vis-a-vis the one for cell type, the tissues tissue level inference
Yeah.
compared, it was robust compared to that of cell type inference. And one would wonder why the one for tissue is robust. I think
Uh,
you hinted about it, but
yes, so one of the main issues that I said before is the convolution is quite complex. And in the cell type level, we have hundreds of potential categories, potential contributors, potential cell types. Where in the tissue level, we only have 30 potential categories. So essentially, the features or the signatures that the convolution will depend on, I think it's much more milder in the tissue level. So I think that's why the performance is better. And you also have cell types can be more transcriptionally similar, especially for different immune cell types. So I think that's another reason why we see that.
okay. The next question is about the Bayes prism, and my assumption is it's a Bayesian approach, and it is consistently performing better compared to sort of regression-based approaches. And is there an explanation for that specifically?
It should be. I don't have any specific explanation for that. It's probably the model architecture and how it tries to understand the different errors and the different contributions. It's just
Okay.
a different algorithmic framework for this problem, I would say.
Okay. I think in conjunction with that, we tend to think about biological data in a way as high dimensional data.
Mhm.
And just out of curiosity, what would you say about the dimensionality of the tissue cell, but also of the cell type in terms of dimensionality just? And the reason why I'm asking that is these days I'm seeing research in the direction of implicit manifolds in sort of diffusion processes. And one of the key interests there is modeling data at a near low dimension. But just to go back, what would you say about the dimensionality when it comes to the cell type and the tissue type?
I mean, dimensionality reduction will be much more complex in the cell types, again, because of how similar and uh, how many more transcriptionally similar are. We try to do one kind of dimensionality reduction. So essentially, we try to integrate the data of the brain with the tabula sapiens. But one of the ways that dimensionality reduction works in biology, or what I research on, what I was trying to find a solution to, it was essentially using common anchors between the data to make it much more. And that's one of the dimensionality reductions that is used in the convolutions. But in our case, because of it was two complete different atlases and with different samples, different patients, different tissues, it was not really possible to do that. So that again, in theory, could also improve the convolution in the future if data sets become available that could allow dimensionality reduction.
Okay. You talked about noise And I think still it is, I think, tissue type that is, I think, robust to noise. My curiosity is about in a simulation setting like this one. Of course, I know you tested on real data, but in simulation, what are the noise sources? Like, where is this noise coming from? And um, how is it being like sort of thought about on a real data set? Like, what are those noise or possible noise sources in the data set? How would they look like?
You mean, what do you mean the structure of the noise, like if it's a log or negative binomial, or do you mean the sources of noise?
No, I mean like the sources of noise. Like when you look at the data representation itself,
Mhm.
uh, what would noise be like within the data, but also within the simulation itself as you're carrying out the simulation. And somewhere I think you hinted on tissue uh, tissue 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 level inference being um, like robust still to noise. And my thinking is, I'm wondering, like, what does noise look like essentially in this setting?
Like practically, you mean in practical terms, what can introduce the noise?
Exactly, yes.
Yes. Eh, that first of all, you have the patient variability. So there is the
Wow.
expression difference between each person. So that's another thing. There is studies now to into EQTLs. Essentially, it's how genes are differently expressed between different individuals. So that's one aspect. Then the other aspect is the size of the tissue. So
Yes.
essentially, we wanted to see one of the reasons we added the noise is to see if the presence of specific tissues, such as if a simulation contain brain, for instance, so that tissue, brain in this example, does not act as a sink of noise, and because of its presence, the noise is reduced. So that's another aspect which is also linked to the size of the tissue in the human bodies, which is also different between individuals. And then you have the aspect of decretation that can also introduce additional noise, and not only decretation, but also the experimental, the wet lab component, the generating the libraries. How a laboratory generates the libraries, which kits they use, which extraction they use, which library prep they use. And it's another aspect of how noise is introduced. And one very important one is how they collected the sample. So there are recent publications in Nature that they show that the collection tube, the blood collection tube that is used in self-RNA studies, can have a big impact on the the data that we get. There is some biases essentially.
Wow. Okay. Interesting discussion, Alex. The members on the call, those who have remained, I don't know whether anyone has a question. But if no one has a question, allow me end the call. We have approached the top of the hour.
Mhm.
Uh, and I want to thank Alex for taking off his time and share this novel work with us. Thank you very much, Alex, and we really appreciate this endeavor.
Thank
And
you for having me, and all the best, and nice to meet you as well.
thank you very much. Goodbye to you, Alex, and goodbye to everyone on the call. See you in our subsequent calls.
See you.
All right.